But today, we really need to get through this material, so we're actually going to keep working on it. We had left off yesterday sorting between the differences between homology and analogy, correct?
Yes.
Remember that homology was when there was a relatedness in structure, a similar structure due to evolution. Analogy was when you had similar structures, but the similar structures were not evolutionarily connected, right? Like the brain of the octopus and that of the human, correct? Now we said that, like, what is the ultimate source? Like, what is the absolute best trick in the book as far as learning about phylogeny or doing phylogeny? Yeah, molecular 100%. Not just genetics, because that's just the gene coding region. All of the molecular part, right? Which would be like everything, every piece of DNA. Let's talk about shared characteristics. And what we're going to do is we're actually going to go back into morphology a little bit. This pin's dead. And the reason we're going to get into morphology again is because this has kind of been the classic way to do it. And also, we really don't have the materials that would do DNA sequencing. Cool? The first thing I think we should mention is that homologies, this pin's dead too. Homologies can help infer phylogeny. Now, again, what was homology? Just to make sure that we've got that 100%. Okay. Similar structures. Right. That are linked through ancestry. So there's a relatedness between those structures. The term cladistics refers to groups of organisms, refers to grouping organisms, I should say, by common descent. So it is the process of grouping organisms together that have a relatedness. From that you form clades, which are when you have groups of species that include ancestral species, or I should say the ancestral species. Clade? Sorry about that D looking. That was my bad. There is a space there, but it's like I can tell you can't see it, and that's my fault. But it includes the ancestral species and all descendants. I'm going to get you to draw this little guy in there real quick. And I want to focus on what that's saying. These are different clades. Okay. Actually, let me back that off before you write that down. Yeah, I'll talk about why there's a shaded part. Maybe you might want to just like do like a box in or like a circle around it. Cool. I think we have three different things going on here, like three different groupings. So I'm going to get you to write down below each of these: monophyletic, paraphyletic, and polyphyletic. The term monophyletic means that you have a group of organisms that includes the common ancestor and all descendants. Now, if you'll notice what you've circled in right up here, you have this common ancestor, right? Your CA, your common ancestor. And you do have all the descendants that you see right through there. Cool? That's what makes this monophyletic. Now, this paraphyletic means that you have the ancestral species, or an ancestral species. So this includes an ancestral species, but only some of the descendants. What's the ancestral species here? Where should I put my dot for my CA? Very, very bottom, right? That's my ancestral species. So we got the common ancestor. Now, what are all the descendants of this common ancestor that I've drawn this bullet point on? Everything. Have I drawn a circle around everything, though? Only around part of that, which means I do not include all of the descendants, only some of the descendants. Now, on this one, you have polyphyletic, which means you have different species, but it lacks the common ancestor of those species. Cool? And you can see there that these creatures that are circled, what's the most common ancestor? This one. Is it included? Polyphyletic group. Me?
Yeah. For the paraphyletic, I really don't understand how the whole thing can be circled.
No, well, because it says you only are including some descendants.
I mean, like,
so this could be paraphyletic if I did him, or if I circle him through him, but as long as you leave one of the descendants out, it's paraphyletic. As long as one descendant is left out.
Yeah, that's how I don't get it.
It's just like when you're grouping organisms, it's all about like when you're creating these phylogenetic trees, it's just like terminology of a phylogenetic tree. Like monophyletic would mean that you include all descendants and the common ancestor of all those descendants. Paraphyletic means that you've included just a couple of descendants and not in a common ancestor, but not all descendants. And polyphyletic is only descendants, no common ancestor. It's just, to me, it's just journal terminology that you use with it. It's all about like mechanisms of just grouping species. I mean, it's just like it's really hard to describe like why you would use this or like an application of it, but I mean, if we were going to create a phylogenetic tree like based on all the creatures that are back there, in a lot of cases we would probably be we would probably be polyphyletic at best. Because we would be including a lot of the descendants of common ancestors, but we don't have the common ancestor back there.
So it's just like these are created based on like what you have present.
Yeah.
Is there one that encompasses like all of them?
If I had a bunch of jars. If I had like
is there like a term that like
oh,
the whole thing was shaped?
monophyletic would be the entire thing.
Okay,
If
so
you think
that-
about it. That would be everything. Like a phylogenetic tree would include everything. The common ancestor and all descendants. And of course monophyletic could just be referring to a single piece of this where you have a common ancestor and all descendants. Now this next part was an AP test question like 2 years ago, so this is maybe unlikely to be asked, but I still think we kind of need to know it. So here's what we're going to do. I'm going to copy, let's see. Get you to copy down another chart real quick. Might want to draw like a little box, like a little table, as in Microsoft Word table. Like about a 5 by 6, 6 columns, 5 rows. Something that would look like that.
A little one?
Yeah, I mean, maybe about, it's pretty small because we just need like check marks in it.
Oh.
And I'm going to write some things off to the side. Backbone. Jaw. 4 legs. Egg. A shelled egg, I should say. And hair. And let's see, we got a couple different organisms here. We've got a lancelet. Very, very end. Primitive chordate. What else do we have? Lamprey. We've got one of those in the back. They look pretty disgusting if you've ever seen one before. There's a tuna. Looks like a lizard of some sort. Or salamander. There's a turtle. And let's go leopard. A cat. I'll let you copy that down. And yeah.
What is that first word on the top?
Lancelet. Lancelet. Let's say another 3 seconds. All done? Nope, okay. More time. We good now? Good. Okay. Now we just have to fill in the blanks, I think. Let's see. Check, 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 check. Tuna, they have jaws. Let's see. Jaw for everyone onward than tuna, I think. Yeah. 4 legs. Tuna don't have legs. Shell. Like that. And then here, leopard only. Kind of works out that it was designed like that. Because I think you would just put zeros or knots, which means that it's totally not having that structure. As you can see, we're basing this off of morphology. And so from this, you would actually create this phylogenetic tree. And if you haven't drawn this in, you might want to draw it in real quick. You have your lancelet. There's your lamprey. There's your tuna. The salamander. The turtle. And the leopard. And you'll see that each division is based on like having this new feature. For instance, what separates the lancelet from the lamprey is just the single. What?
How does the leopard have a shell of eggs?
What do you mean?
You have the check mark on shell egg for leopard.
It just means that it's an amniotic egg. It's not like a.
I was thinking like.
You're not like. Don't think of like. No. Think of like an amniotic egg, which means that it's an egg that is encased in something. Amniotic. So the difference between the lancelet and the lamprey is what? What's like the one feature that's unique between lamprey that is not like present in the lancelet? Okay, so obviously right here you have to have backbone as the breakup here. And the breakup here would be the jaw, the legs. The difference here is going to be the amniotic egg. And then here, of course, it would be here. So when you're creating a phylogenetic tree, really the difference between your creatures is like all of these features, obviously. And these features actually could create, like, could help you out, I guess you could say. Like if I had a creature that was lacking a backbone and I wanted to fit it into all this, where would I stick it? If it was lacking a backbone, I could not put it beyond this line. It would have to be coming off of what little bit I have here, or off of the lancelet line, depending on how it's related to the lancelet. You okay? Okay. Good? I would say that we have an outgroup here. I would say that the lancelet is the outgroup. Which has no features in common. But is related to everything else. For instance, the lancelet doesn't have any features in common, but is a chordate just like the rest of these. They're in the same phylum. And of course, what would be like the most, what would be a better mechanism of putting these in order or in a phylogenetic tree? Using their DNA, using molecular evidence, as opposed to using morphological evidence, right? And you can see that this is a pretty simple concept to do, but this would be simple because you're looking at like creatures that are pretty much like very, very, very different, correct? If you were starting to try to stick like all the different types of like cat creatures, if you will, or all the like different reptiles or all the different amphibians, you would find it would be quite difficult to like piece together one that's like that based on just morphology, which is why you would use molecular evidence first and foremost. Cool? I'm going to show you this picture and then we're going to move on to the next chapter. I'll let you look at that real quick. Might even want to take some notes or jot that down.
The whole thing.
I would say really just the chart up top could help, possibly. Now, that chart up top could create this down here. I mean, it would be pretty simple to draw this little phylogenetic tree, or the two possible ones. You actually see that the first tree, the one on the left, is more likely, which actually states that we're more related to a fungus than a fungus is to a plant. Seems a little illogical because you would put the fungus and plant as being pretty closely related, but they are far apart. I just think this is important to kind of see the differences between creatures and how it's kind of perplexing that we are more like a fungus than a fungus is a plant. Okay, all done? Let's say another 3 seconds. Sure.
What is it the tree do?
It's the different ways that you can take that data and extrapolate like relatedness. To create that, like all that data right there could give you 2 possible trees, but the first tree is just a more likely event. And honestly, it's because it keeps these differences that you see right there about the same. These differences that you see right there are, to me, a little too much. Right? I mean, the separation here should not be that degree. It would make better sense that there was this much separation. Everyone seems tired. What happened?
0 hours.
0 hour? Stings. What happened? What happened? 2 practices? For what? That's awesome.
I started my homework at 11.
What were you doing?
Well,
Procrastinating.
I was, what?
Procrastinating.
No, I was manning, and then we had an SOS meeting. It was really good.
SOS? Well, let's move on. Hopefully, I'm sorry about that. Let's talk about the history of this planet. And like how life has evolved from then. Of course, you would know it's tons of macroevolution, right? Sprinkled in along the way with these catastrophic events. Your book outlines maybe 6 or 7 possible major events that have happened, which would include the asteroid hitting the planet Earth or the meteor, whatever it is, and destroying all the dinosaurs. So you're looking at a bunch of different events that have happened, or at least several major events that have happened that have led to global change of biological creatures. And remember that things move pretty steadily and very little between those time frames, but then all of a sudden there's tons of evolution. That's a punctuated equilibrium model. Cool? But I think before we talk about, instead of just getting into like all the major changes that have happened, let's talk about how life could have started. Like what would make that possible? And to me, there's 4 things that you have to have happen. The first would be the abiotic synthesis. Of organic molecules. Now, what does abiotic mean? Okay, so the non-living making of organic molecules. Now, hopefully you've picked up along the way that organic molecules are made only in 2 mechanisms. They're made by biological systems. Can someone give me an example of a biological system? You. Your cat. All those creatures that I have in the back that are sitting in the formaldehyde, they used to be able to. Not now, but they did once. Right? They were all biological systems. You can make organic molecules, right? Urea, CO2, anything that has carbon, you make it. You're making organic molecules. What's the other way we've learned to make organic molecules? In a chemical setting, in chemistry. Pharmaceuticals are like a great example, right? Medications, those are all organic molecules most of the time. I think they almost virtually all are. right? So we have to think about this. You guys, you all know what the Big Bang Theory is, right? Not this TV show that's out there. I'm talking about the real thing. There was this Big Bang that happened, and all the atoms that were in this area became this planet, right? The planet's made of rock and about nothing. And now here we are today. So the first thing that would have to happen is you've got to create these organic molecules, life molecules, but you do it from nothing. Make sense? Well, at least you understand what it's saying. You have to start making the chemicals that would make a cell first. The second thing you do is you take these small molecules of these small molecules to form larger ones, and what would we call the larger molecules? Macromolecules or polymers, right? Polymerization has to happen. Now, what are some examples of macromolecules?
Four that we have now.
Okay, which are what? I want someone to give me one, but not you. That's one. Someone totally different, give me another one. Proteins, a totally different person. Lipids, someone else. Okay, nucleic acids. There's your four, right? Or major groups of macromolecules. Those came into existence by the random assemblage of these smaller organic molecules. And then after that happened, there had to be a packaging of these molecules. Into protobionts. What a protobiont is this is basically like a prototype cell. What's a prototype?
Like a model.
It's a model, not the real thing, but close to what the real thing's gonna look like. So these are the first types of cells that exist. Like an almost cell, if you will. And then the last thing that would have to happen is you would have to have this new ability, so a gained ability to self-replicate organic molecules. Can you give me an example of a molecule that you replicate in your cells? If you've already answered a question, I don't want to hear from you. What's a molecule that you replicate yourself? Okay. DNA. You replicate your DNA, correct? Can someone answer totally different why I would replicate my DNA? Why do I replicate DNA? New cells, right. Why would you need new cells? Repair, to live, growth, right? Okay. So you see this process that has to happen, right? The random making of these molecules, and they have to come together to form something large. Like, all of a sudden, I want you to picture this in your head. This is like how it would go. All of a sudden, a nucleotide showed up on this planet Earth, primitive Earth. There was a nucleotide that was randomly formed, or an amino acid randomly formed. They sit there for I don't know how many hundreds of years, until there's enough of these nucleotides or amino acids that they come together to form proteins and DNA. Then, for some reason, in a crazy world that this planet was, I guess at the time, those little macromolecules all came together. How, I don't know. They did. And they got packaged into a membraned type structure, kind of like a cell. Then some time elapsed and they gained an ability to reproduce. So you take it for whatever it's worth, I guess. But that's the way it would have to go. Cool? Now, you'd be like, well, that seems like way too unlikely to happen. So there's the contradiction, of course, that would exist between like science and then religion. I will say that at the very least, step one has been facilitated in a synthetic setting. The Miller-Urey experiment gave proof of step 1, which was the making of organic molecules from a primitive Earth environment. They basically made in their lab, they created a little setting that in a test tube, or actually it was a huge beaker, a setting that had the same gases in composition and the same like electrical forces, like lightning storms and everything else that primitive Earth had. So they made basically baby Earth in a test tube, and they let it run for I don't know how long. And one day they came in, they found that the baby Earth inside of their test tube had made randomly some organic molecules. So it was possible that organic molecules could just occur, could be formed in a setting like that. Right? Now you could think about the stats on how much, what's the likelihood of this and this and this and this happening, and here we are today to discuss it. And that's kind of, I think, to me, that's a little bit more. This statement is a little bit more contradictive than evolution is to religion, because evolution just gives a process, but this actually would completely contradict like religion. So I think the decision is based on this. As far as what early Earth contained, I think you do need to know that it contained tons of nitrogen, so N2 diatomic, nitrogen oxides NO2, NO3, carbon dioxide, methane CH4, ammonia NH3, hydrogen H2, and hydrogen sulfide, which is what little egg bombs smell like. Eggs, H2S. Most of the time you could actually assume that these chemicals right here are from volcanic activity. So think of an electrical storm, volcanoes everywhere, these chemicals are flying through the atmosphere, and somehow they assembled into amino acids, nucleotides, fats, carbohydrates.
So there are many Earth created those.
Their test tube, in their test tube Earth, they put these chemicals, they set up some lightning storms like with just an electrode. They also set up like the right conditions of heat, and after some period of time, and I couldn't tell you how long it was, but their baby Earth was able to take these guys and assemble a very simple organic molecule. And it's been repeated, so it can happen. I think the thing you have to remember is that if you can make this happen, okay, but then what would be the difficulty of making this happen? How much time would be involved in making this happen? Because this is an assemblage of many of these guys. This planet is huge, right? Let's say that you made two of these little molecules and you want them to come together, but one is on Antarctica and the other one's in Florida. It's a distance. Of course, at the time it was only one big continent, but oh well, there's still distance that separates them. So this is either unlikely or would take tons of time, and that's why biologists would certainly put the age of this Earth like way more than like what other books would read. According to biology or like this theory, the Earth would have to be somewhere around about 4.2 billion years old. 4.2 billion years old, which is like just a couple of 100 million years younger than the sun is, our star. Yeah. Good. You'll notice that primitive Earth is lacking oxygen too. We'll need to talk about how that came into being. Okay. Let's see. Oh, here's something else that's fun too. Take it for what it's worth, but in meteorites, because they hit this planet, I guess to some degree, amino acids have been found in the meteorites. Did y'all know that, or have you heard of this? They've actually found like the same organic molecules that we use in rocks that land on this planet from like the outer space. So I just thought I should throw that in there. Cool. Let's talk about what a protobiont is a little bit deeper. What was a protobiont? Yeah, this was like the primitive cell, right? This would be like an aggregation of abiotically produced molecules. Of course, they are surrounded by a coat or you could say a membrane, kind of one and the same. They have simple reproduction and metabolic systems. Their spontaneous generation has been proven in a lab setting. There have been experiments where they set all these macromolecules inside of the early Earth environment, and the protobionts do form. The problem that they've had and the time it would take, the issue on your list of 1 through 4 from the previous page would be really step 2. How could you get all these little molecules together? How would they know to come together? That's, to me, is like the biggest issue. Cool. And one thing we'll notice that they used, when they first were able to have their genetic material, they used RNA instead of DNA for their genetic material. And that was valuable because RNA, again, did serve as their genetic material. It was their unit of inheritance. But it also can act as an enzyme. Acts in an enzymatic fashion. It's called ribozymes. We've actually seen that before. Can someone name a ribozyme? I would say, you could say ribosome would be partially, but that's made of protein too. Can someone give me an enzymatic type molecule that is made only by RNA?
Oh, the backwards?
Reverse transcriptase?
Yeah.
That's an enzyme. That's a protein. tRNA, right? Carries the amino acids, has a function. It's a doer of a cell. Cool? So tomorrow we'll pick up on protobiomes a little bit more. Try to get through this chapter. If you can come hang out with Mr. Fields today, it would be great. And we'll get some stuff done. Thank you.